Hello, and welcome back to What is a Biology? I'm your host, as always, Mr. Johnny Hopkins, and today we'll be covering genetic vocab. But this is going to be less than regular, normal, random hodgepodge, but rather this is going to be focusing on the structure of chromatins, specifically going into nucleosomes and histones. So we'll discuss histones, the histone code, heterochromatin versus euchromatin, as well as this SMC complex, and just the general chromosomal structure with autosomes, the telomeres, centromeres, kinetochore, just the basic simple stuff. And so all this will come back later in a much more detailed manner with going to very individual specific things. So first of all, what is a histone? And the answer is it's a protein that compacts and organizes DNA. It's made out of a dimer of 4 subunits. So it's a homodimer of 4 subunits. Don't know how the term would be right there. But so this guy helps organize and condense the DNA, because if you remember, DNA displayed all out, end to end, is about 2 meters long in my body for every individual cell, which is huge. So we need to condense it and make sure that fits inside our around 10 micron long cell. So what we do is we use chromatin. We use histones. And so these histones actually make up 50% of essentially the DNA in the cell by mass. So chromatin is just this condensed DNA protein structure that is 50% DNA and 50% these histones. So these guys are very, very important. And so these guys are made up of, so we've got two concentric rings, and there are 5 different types of subunits: H1, H2A, H2B, H3, H4, and all these which can be post-translationally modified. And so these guys kind of just come into a circle like this, and you got H2A, H2B, H3, and H4, and they stack on top of each other. So why is it like this is that you have DNA then come in and it wraps around it 1 2/3 times around, and it, so essentially what that happens is 147 base pairs are on this guy, the histone. And so what we call a structure of DNA on these histones is a nucleosome. It's just the term. The nucleosome is a histone with DNA on it, and it exists in the genome. So then we go into what is a chromatin. And a chromatin is essentially just the zigzag structure of DNA with these histones. And so all you can have is a zigzag pattern where essentially these histones lay right on top of each other, whereas like this is a very much a side view, but when you look over it, it'll just kind of look like it'll just be very much all stacked on top of each other in a very, very organized manner. So that's what the chromosome does, and it's got these stacked nucleosomes. And this chromatin, because it's so compact, it's not transcribed. So what that means is we don't actually look at the genes on there. RNA polymerase cannot attach onto this DNA and make it make an RNA, which can then make a protein. So in fact, what we see usually is how do these guys condense like that versus just kind of stay a little strand outside with just a little bit of histone here and there, a little bit of nucleosome here, a little bit of nucleosome there. And what happens is that the N-terminal tail usually of these histones, the N-terminal, usually the 19 to 30 residues that are extended, as well as the 2A and 2B have flexible C-termini. And these guys can be very much, these guys have a very strong opportunity to be post-translationally modified. And these post-translational modifications cause a lot of different abilities for condensation of the nucleosomes and chromatin structure. So one of them, known as H4, the glycine 16, if you post-translationally modify that can lead to fiber formation. One interesting thing to note as well is that a lot of different experiments can look at H3 by itself, H4 by itself, H2A and H2B by themselves, Or they also typically can look at H2A and H2B together to see how that perturbs the different protein and see how it all interacts as a whole system with these nucleosomes. So, now as I just stated many times before, you've got PTMs, post-translational modifications, and this is part of the histone code. So these PTMs cause different structure. There are many different types of post-translational modifications to any sort of protein, but these are the typical ones that are looked at inside the cell. Acetylation, methylation, phosphorylation, and ubiquitination. Acetylation, you add an acetyl group. Methylation, you add a methyl group. Phosphorylation, you add a phosphate. And ubiquitination, you add a ubiquitin. And if you might remember, ubiquitination usually means you degrade a protein. You add on a ubiquitin to a protein, and then that protein gets signaled for destruction, essentially. That's the ubiquitous way of ubiquitin. And however, though, in this, it's not the case. Ubiquitin does its own different job, which we'll get into many lessons down the road. So, the typical one is acetylation on lysines in any of these guys usually. And when you acetylate them, you neutralize the charge of this. And then you actually reduce the condensation. They get further and further apart. So when you reduce these, when you acetylate, that's what happens. And so what typically you get is two different types of structures, heterochromatin and euchromatin. Heterochromatin is highly condensed DNA, and this is not usually actually fully decondensed during the mitotic process. And so it looks kind of like this. You got DNA in, you got DNA out, and just a bunch of histones all around. You can't access any of it. And so one thing to note is that this is very common in something like in the X chromosome in females, because females have two X chromosomes, and to try to avoid overexpressing different genes, they actually make one of them heterochromatin and don't let it get fully decondensed. So we actually just see one X chromosome. And that would be something called an epigenetic factor of which X is fully condensed. And there's other parts of the human body that we'll get into later on that can be highly condensed. And this is the epigenetics that people talk about, trying to understand and help improve our genetic code, or what is expressed in our genes by removing the nucleosomes or adding nucleosomes so we don't overexpress or express a certain protein. And then we have euchromatin, which is the opposite of heterochromatin, essentially. You've just got a little bit of, it's like beads on a string is what they say. It's just kind of DNA, histone, DNA, histone, DNA, histone. So you can pretty much get anything to attach to it and make your RNA or protein from this basic guy. And so one type of protein that actually attaches something that has a bromodomain. And a bromodomain is just a protein domain that recognizes acetylated histone tails. And it's usually done in euchromatin because, as previously mentioned, acetylation happens on the reduced condensated ones, and euchromatin is the only ones that you can actually grab onto. So these guys are typically acetylated in their histones. So continuing on, there's another organizational stuff of the cell, and one of them is called the structural maintenance of the chromosome complex, SMC. And what these guys essentially do is just wrap a ring around DNA and just keep them topologically linked nearby. So there's two different typical ways, is that essentially you have one strand over here, one strand over here, then just like the SMC is like, hey, these guys should have a very similar purpose, and we should, whenever I want to look at this guy, we should also look at this guy at the same time, so it just kind of links it. It wraps around both of them, like that. And then you also have the other one where essentially a loop extrusion, where we'll just take a flat DNA and then kind of pull it together into making a loop. So like that, yeah, like that. And so that's what happens over here. And so these helps different organization of the cell for different 3D topological purposes. Now, moving on, when we have all these histones and SMCs together, we can get these highly organized structures known as chromosomes. And chromosomes don't exist in the cell natively, because typically, natively, we are an interphase. Interphase is when essentially the cell is not worried about reproducing and dividing and trying to make more of itself, but rather it's just trying to live and stay alive just keep and maintain a nice homeostasis. So, so we have all these chromosomes, but they typically only appear during mitosis as a highly condensed structure. Otherwise, they are these sprawling entities like that have a little bit of heterochromatin, a little bit of euchromatin over here and over there. And so you've got a karyotype, which is just the collection of chromosomes. It's just all the chromosomes together of a single cell. And then you have autosomes, which are the chromosomes that are not sex-linked. So everything that's not X and Y in the human body. You have synteny, which is essentially just when you have like two different genes in appear in the same order, say gene A and gene B on chromosomes are expressed from gene A to gene B in chromosome 5, and then have gene A and then gene B in chromosome 7, and then those are actually the same gene on two different chromosomes, and that's synteny, and because they're in the same order as well. You have something called autosomally replicating sequence, which essentially in the eukaryotes, this ARS, acts as a 100 base pair, like origin of replication for reproducing these these chromosomes. And so going on, we'll talk about, so the centromere is this guy. It's the center essentially of this chromosomal structure. So that's what a chromosome looks like. And you have a centromere in the middle. You have a chromatid, which is one half of this, which gets pulled in the mitotic spindles. And then you have a telomere at the end, which we'll get into in a second. So a kinetochore is, a centrosome. So that center guy has 3 regions. It's got the region 1, region 2, and region 3. Who would have thought region 1, region 2, and region 3 would have been the names for the 3 regions? It's by that or A, B, and C. It's a coin flip. And so we have the centrosome. There are 3 regions. 1 and 3 are very well conserved, and they bind to microtubules. These guys actually have very different lengths as well, whereas the 2 in all eukaryotes the region 2 has a very constant length that's AT rich. And so this centrosome increases and decreases essentially based on the size of the eukaryote, with humans being significantly larger than yeast. And so this 1 and 3 are what binds to the microtubules. So what the heck is a microtubule, man? And so a microtubule in the mitotic cycle, you have so you've got a chromosome, and we have our cell. And so what happens is that these MHC complexes, essentially we have mitosis happen, is that these mitotic spindles, these guys, actually pull apart this chromosome. So we've got one half of the chromatid on one side, one half on the other. So that's what happens during mitosis, is these mitotic spindles are pulling the chromosomes apart. And so one thing to note is that this part of the chromosome is the same exact thing as this part of the chromosome. They're exact replicas of the same thing. They're the same sequence. And because you just reproduce it, and then you pull it apart, so you can make 2 cells with the same exact DNA. So that's the point of that. And so the kinetochore is essentially this protein structure that associates at the centrosome that is able to associate with these mitotic spindles. So the kinetochore essentially is the centrosome. It associates with this 1 and 3 regions. Now, continuing on, I mentioned this thing about telomeres at the end. So when we reproduce in our bodies, DNA has a leading and lagging strand. If you might remember with the Okazaki fragments, by going backwards, we actually don't see, we actually see that we lose a couple of base pairs every single time we replicate our DNA. And so what happens is that we have something called telomerase, which actually can sometimes increase the size of this lagging strand and make so we have longer strands at the end of our DNA. However, that's not always occurring in our autosomal cells. So what happens is that telomere is this repeat of TTAGGG at the end of our DNA that's over 1,000 base pairs long in vertebrates. And this actually allows us to just not lose any information every single time that we reproduce ourselves in our own human bodies. So telomerase is the enzyme that typically works to try to keep these guys longer and stay long, so we don't pass any genetic, we don't shrink the size of the genome every single time we reproduce. And so that's what the job of telomerase is. However, and there was an experiment where they looked at mice where they removed telomerase. And what they saw is pretty much the same exact viable systems, viable mice from generation 1 to generation 2. They're pretty much the exact same mice, no difference. After you start to see a little bit of adverse effects where you have younger, they die earlier, as well as they aren't able to reproduce at all. This starts to exponentially increase essentially, and then there's no ability to have any offspring after 6 because the genes are too messed up effectively. So that is everything I wanted to cover today. So just remember, histones are what helps organize the cell's DNA, and they make these nucleosome structures that help organize it and can do epigenetic factors and highly condense it into heterochromatin, or just be a pretty open euchromatin, so it's going to be actively transcribed. You have SMCs that help organize the DNA by topologically linking to different parts. And then you have chromosomes that have the structure of the centrosome in the middle, telomeres at the end, and chromatin are each individual part that gets pulled inside inside the mitotic cycle, and so with the chromatids, they're exact replicas of each other, so we can reproduce and have that. And these chromosomes do not exist naturally during interphase, but rather show up only during mitosis. So that is everything about genetic vocab in general, and next time we'll cover something completely different about post-translation, about just transcriptional control with something called transcription factors, which will be covered and talked about pretty much every single day after this. So that's all. I've been your host as always, Mr. John Hopkins. Thank you for watching.